This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. Today, we're traveling back 7 million years to meet one of humanity's most ancient relatives, Sahelanthropus. Discovered in the deserts of Chad, this remarkable fossil has sparked heated debates about our evolutionary origins and challenged everything we thought we knew about early human ancestors. In this narration, we'll explore the story of how a single skull has reshaped our understanding of when and where our journey began, examine the controversy surrounding its place in our family tree, and discover what this ancient find reveals about the path from ape to human. Chapter 1. The Discovery of Toumaï. On July 19, 2001, beneath the scorching sun of the Jurab Desert in northern Chad, a Chadian researcher named Ahonta Jimdamalbai made a discovery that would ignite decades of scientific controversy. Walking across the wind-scoured landscape of a fossil-rich area called Toros Manila, Jimdamalbai spotted something extraordinary jutting from the ancient sediments. It was a skull, darkened by time and mineral deposits, giving it an almost charred appearance. A crust of black mineral deposits covered much of the surface, lending it what one observer would later describe as a slightly malevolent look. But this was no ordinary fossil. What Jimdamalbai had found would be proclaimed as nothing less than the oldest known ancestor of humankind. Jimdamalbai was not alone that day. The discovery team included 3 other members: Fanone Gongdebe, Mohamed Adoum, and Alain Beauvillet, a French geographer who had organized this particular prospecting mission. They were part of the Franco-Chadian Paleoanthropological Mission, known by its French acronym MPFT, a collaboration bringing together researchers from the University of Poitiers in France, the University of N'Djamena in Chad, and Chad's National Research Support Center. Leading this ambitious venture was Michel Brunet, a French paleontologist who had spent years searching the fossil beds of Chad for evidence of ancient human relatives. The skull they found was remarkably complete, though it had suffered the indignities of deep time. Millions of years of geological pressure had fractured it, displaced fragments, and plastically deformed the bone. The left side was crushed inward, and the entire structure was compressed from top to bottom. But even in its distorted state, the skull's significance was immediately apparent. At 7 million years old, it would push the human lineage far deeper into the past than most researchers had imagined possible. The skull would soon be named Sahelanthropus tchadensis, but it would acquire a more intimate name as well: Toumaï, meaning hope of life in the local Daza language. This name carried profound significance in the region, traditionally given to children born at the edge of the dry season, when their chances of survival hung in the balance. The name was chosen by Idriss Déby, then president of Chad, partly to celebrate the memory of one of his comrades in arms who had died in the country's long civil conflicts. It captured both the fragility and the promise embodied in this ancient remnant of our possible past. When Brunet brought the skull back to France, he understood the theatrical power of the moment. Summoning his colleagues at the University of Poitiers into a classroom, he held up the fossil and asked with deliberate drama, "What is this thing?" The performance was characteristic of Brunet's approach to science. He was not just a researcher, but a showman who understood that controlling the narrative around a discovery was nearly as important as making the discovery itself. Between July 2001 and March 2002, the team recovered additional fragments from the same general area. In total, they found 9 cranial specimens representing at least 6 different individuals. These included not just the famous Toumaï skull, designated TM266010601, but also several pieces of jaw, including fragments with tooth sockets and several isolated teeth. The fossil-rich sediments at Toros Manala preserved not just Sahelanthropus remains, but also evidence of ancient lakes and forests, a far cry from today's harsh desert, suggesting a complex environment millions of years ago. What made the Toumaï skull particularly valuable was its state of preservation. Despite the deformation, it was one of the most complete skulls ever found from this incredibly ancient time period. Previous discoveries of potential early hominids had often consisted of fragmentary jaws or isolated teeth. Here, researchers had nearly an entire cranium to work with, offering an unprecedented window into what our earliest ancestors might have looked like. The dating of these fossils presented its own challenges. The Jurab Desert lacks the convenient volcanic ash layers that have made dating so reliable at sites in East Africa. Instead, the team initially relied on biostratigraphy, comparing the other animal fossils found at the site with similar species that had been reliably dated elsewhere. This approach suggested an age of between 6 and 7 million years. Later, in 2008, researchers applied a more sophisticated technique using isotopes of beryllium, refining the age estimate to between 7.2 and 6.8 million years old. The badly crushed skull would later require sophisticated CT scanning and digital reconstruction to properly analyze its features. The reconstructed skull revealed a fascinating mosaic of features. The brain case was small, with a cranial capacity estimated at between 360 and 370 cubic centimeters, similar to the brain size of a modern chimpanzee, and roughly 1/4 the size of a modern human brain. The skull also had pronounced brow ridges, projecting forward above the eye sockets in a manner reminiscent of apes. But other features seemed more human-like. The face was relatively flat compared to living apes, without the protruding jaws and snouts typical of chimpanzees and gorillas. The canine teeth, while robust, were notably small for a male ape, lacking the dagger-like projections that male chimpanzees use in displays of aggression and competition. The position of the foramen magnum, the large opening at the base of the skull where the spinal cord exits the brain, was also notable. Its placement seemed to suggest that this creature held its head atop an upright spine, rather than thrust forward in front of the body. On July 11, 2002, Michel Brunet and his colleagues published their findings in the prestigious journal Nature. The paper made bold claims. They proposed that Sahelanthropus tchadensis represented a new species of hominid, and moreover, that it was the oldest and most primitive known member of the human family tree. The genus name honored the Sahel region of Africa, the semi-arid zone south of the Sahara where the fossils were found. The species name, tchadensis, honored the nation of Chad. But Brunet and his team did more than simply publish in an academic journal. They understood the profound significance of their discovery and announced it to the world with appropriate fanfare. In the Chadian capital of N'Djamena, Brunet presented the findings before government officials and television cameras. His words were dramatic and unequivocal. "A new hominid is born," he declared. "By virtue of his age, he is the ancestor of all Chadians, but also the ancestor of the whole of humankind." This was an extraordinary claim. The Chad location, west of East Africa's Great Rift Valley, where most early human fossils had been found, immediately challenged prevailing theories about human origins. At 7 million years old, Sahelanthropus predated other claimed early hominids by as much as 1 million years. Previously, the oldest widely accepted hominid had been Orarin tchadensis from Kenya, dated to about 6 million years ago. There was also Ardipithecus kadaba from Ethiopia, dated to about 5.8 million years ago. Sahelanthropus, if correctly identified as a hominid, pushed the origin of our lineage significantly deeper into the past. Brunet emphasized several features of the skull that, in his view, clearly placed Sahelanthropus on the human side of the divide from other apes. The reduced canine teeth suggested a shift away from the aggressive male-dominant social structures typical of modern apes. The flat face and the position of the foramen magnum hinted at adaptations toward bipedalism. The thickened tooth enamel suggested a diet that included harder foods than the fruit-heavy diet of modern chimpanzees, perhaps indicating a creature adapting to more varied food sources. The name Toumaï quickly captured public imagination. Here was not just an abstract scientific designation, but a name that evoked the precarious hope embodied in a child born at the harshest time of year. The metaphor resonated. This ancient skull represented our species' own tenuous beginnings, a hope of life that somehow persisted against the odds through millions of years of evolution. For Chad, the discovery became a source of immense national pride. Toumaï appeared on Chadian stamps and currency. The country's national airline was renamed in honor of the fossil. In a nation that had endured decades of civil war and instability, Toumaï offered something positive, a claim to a unique place in the human story. Here, in the heart of Africa, might lie the very roots of all humanity. The scientific community reacted with a mixture of excitement and skepticism. The excitement was understandable. A 7-million-year-old skull in excellent condition was an extraordinary find, regardless of exactly how it fit into the human family tree. But the bold claim that this was definitively the oldest human ancestor drew immediate scrutiny. Identifying whether a fossil ape is a human ancestor, a chimpanzee ancestor, or an ancestor of both requires careful analysis of numerous anatomical features, and reasonable experts can disagree about the interpretation. The holotype specimen TM26601-0601, the Toumaï skull, became the foundation for an entirely new genus and species. But a genus and species based primarily on a single, distorted skull is inherently provisional. In the careful language of science, Sahelanthropus tchadensis was proposed as a hominid, as the oldest known member of the human lineage. But proposals require testing. They require additional evidence, further discoveries, and rigorous peer review. The foramen magnum position, while suggestive, was not definitive proof of bipedalism. The orientation of this opening can be affected by many factors beyond locomotion, including the length of the face and the angle of the skull base. What was really needed was postcranial material, bones from below the skull. In modern humans, our adaptation to bipedalism is written throughout our skeleton, in the shape of the pelvis, the angle of the knee joint, the structure of the foot. Without these bones, claims about how Sahelanthropus moved remain speculative. The skull could tell researchers much about brain size, diet, and potentially about head posture. But the skeleton would have been far more informative about the crucial question of whether this creature walked upright. This limitation would later prove significant, setting the stage for one of the most bitter controversies in modern paleoanthropology. But in 2002, as the world marveled at the 7-million-year-old skull from Chad, the scientific community had been presented with a bold hypothesis and a spectacular fossil. The stage was set, the claims were made, and the fossil was there for all to see. Even as Brunet made his proclamation about the oldest human ancestor, skeptics were already questioning whether a distorted skull alone could prove bipedalism, or hominin status at all. Chapter 2. The Bone That Divided Science. The announcement of Sahelanthropus tchadensis as the oldest human ancestor rested heavily on a single crucial claim, that this 7-million-year-old ape walked upright. Bipedalism, the ability to walk on two legs, is the defining characteristic that separates the human lineage from other apes. Modern chimpanzees and gorillas can walk upright for short distances, but they are fundamentally quadrupedal, moving on all fours with their knuckles supporting the front of their bodies. Early human ancestors, by contrast, evolved skeletal adaptations that made upright walking their primary mode of locomotion. This shift freed the hands for carrying objects and eventually for making tools. It changed how early hominids interacted with their environment and may have been the spark that set our lineage on its unique evolutionary path. For Sahelanthropus, the evidence for bipedalism came initially from the skull alone. specifically from the position and orientation of the foramen magnum, that large opening at the base of the skull where the spinal cord connects to the brain. In Michel Brunet's 2002 description of Toumaï, this feature took center stage. In quadrupedal apes, the foramen magnum is positioned toward the rear of the skull base because the head projects forward from a horizontal spine. In bipedal humans, the foramen magnum sits directly underneath the skull because the head balances atop a vertical spine. The foramen magnum in Sahelanthropus appeared to be positioned forward of where it sits in modern apes, suggesting that the skull was carried atop an upright body. Brunet and his colleagues considered this evidence compelling. The forward placement and downward orientation of the foramen magnum indicated, in their view, a mode of locomotion on two legs. Combined with other anatomical details, they classified Sahelanthropus within Hominina, the group that includes humans and our direct ancestors after the split from chimpanzees. But almost immediately, skeptics raised objections. The problem was that the position of the foramen magnum is not an entirely reliable indicator of habitual posture. The opening's location can be influenced by many factors beyond locomotion. The length and angle of the face, the flexion of the skull base, and the overall shape of the cranium all affect where the foramen magnum ends up. Moreover, the Toumaï skull had been severely distorted by millions of years of geological pressure. Even with sophisticated computer reconstruction, could researchers be certain they had correctly determined the original position of the foramen magnum? In 2005, a team led by Christoph Zalikofer published their virtual reconstruction of the Toumaï skull in Nature. Using high-resolution CT scans, they had digitally separated the fractured pieces, removed distortions, and reassembled the skull into what they believed was its original configuration. Their reconstruction supported Brunet's interpretation. The reconstructed foramen magnum was indeed positioned in a manner consistent with upright head carriage. Not everyone was convinced. Critics questioned whether the foramen magnum position really proved anything about bipedalism. The distortion of the skull made any reconstruction uncertain. And even if Sahelanthropus held its head upright, this did not necessarily mean it walked on two legs habitually. Many animals hold their heads upright while remaining quadrupedal. What the scientific community really needed was not more debate about the skull, but evidence from the rest of the skeleton. The pelvis, the femur, the foot bones, these would provide direct evidence of how Sahelanthropus moved. A bipedal pelvis has a distinctive shape, broad and basin-like, to support the internal organs when standing upright. A bipedal femur angles inward from the hip to the knee, bringing the knees close together underneath the body's center of mass. The foot of a biped has a specialized anatomy, with an arch and a non-grasping big toe aligned with the other toes. In the 2002 Nature paper announcing Sahelanthropus, Brunet and his colleagues explicitly stated that no postcranial material, no bones from below the skull, had been found. This was a significant limitation. Without limb bones, the claim of bipedalism remained tentative, based on a single feature of a distorted skull. But this statement was not entirely accurate. A femur, a thigh bone, had in fact been found at the site in 2001, on the same day as the Toumaï skull. Found lying near the cranium was what appeared to be the shaft of a left femur. However, this bone had been collected along with numerous other animal fossils from the site and shipped back to France mixed in with boxes of fossil fauna. It was catalogued as an undetermined long bone and stored with the animal specimens at the University of Poitiers. For nearly 3 years, this potentially crucial piece of evidence sat unrecognized in storage. Then, in 2004, a graduate student named Ape Debate was conducting a study of Chadian fossils for her master's thesis. Among the specimens was the mysterious long bone. She needed expert guidance to identify what she was seeing. Roberto Macchiarelli, a paleoanthropologist at Poitiers, initially declined to examine the bone. He knew the sensitivities around Chadian fossils and did not want to overstep professional boundaries. But Bergeret was a student seeking help, and Macchiarelli felt he had a duty to assist her. When he looked at the bone, he immediately recognized what it was: the shaft of a primate femur. Checking the inventory records, he discovered it had been collected from the same locality, on the same day as the Toumaï skull. Macchiarelli's excitement quickly turned to apprehension. Examining the morphology, he saw features that troubled him. The curvature and internal structure did not look like what he would expect from a habitual biped. He told Bergeret not to allow the bone to be cut for sampling. This appeared to be the femur of Toumaï, and it needed to be studied as such. When Brunet returned from Chad and learned that Macchiarelli had been examining the femur, he was furious. A laboratory meeting was convened in the presence of a university dean. There was talk of people who take advantage of our being away in the field to rifle through our collections. Brunet spoke of a Judas. The dean wrote that a grave professional offense has been committed. Shortly after, Bergeret's research materials were confiscated. The fossils were being renumbered. The femur disappeared from scientific view. Years passed. Brunet and his team published additional papers on Sahelanthropus, but the femur was not mentioned. Rumors began to circulate. In 2018, the journal Nature published an article titled "Femur Findings Remain a Secret," discussing allegations that a thigh bone existed but had not been described. Macchiarelli and Bergeret spoke on record about what they had seen. The implication was deeply troubling. If the femur contradicted the claim of bipedalism, was it being deliberately suppressed? The controversy simmered for years. Then, in 2020, Macchiarelli and several colleagues published their analysis of the femur in the Journal of Human Evolution. Their conclusion was stark. The morphology of the bone was not consistent with habitual bipedalism. The cross-sectional shape, the distribution of cortical bone, the overall structure, all suggested this was not the femur of a creature that walked upright as its primary mode of locomotion. In 2022, a team led by Guillaume Daver and Frank Guy, both colleagues of Brunet, published their own analysis in Nature. They revealed that not only the femur, but also two partial ulnae, forearm bones, had been recovered from the site in 2001. Their analysis reached the opposite conclusion from Macchiarelli's. The femur, they argued, showed several features consistent with bipedalism. There was a ridge on the bone where the gluteal muscles would have attached, a gluteal tuberosity like that seen in upright walking hominins rather than the spiral pilaster typical of apes. The femur also showed femoral antitorsion, a natural inward twist that helps orient the legs forward when walking upright. But Daver and Guy acknowledged that bipedalism was not the whole story. The ulnae showed features indicating that Sahelanthropus also climbed in trees. The bones had curved shafts and cross-sections that were thickened in the front-to-back dimension, suggesting the forelimbs bore significant weight. The hand bones showed evidence of arboreal clambering, enabled by firm hand grips, clearly differing from the knuckle-walking pattern of gorillas and chimpanzees who lean on the back of their phalanges. Critics pushed back immediately. In 2023, Mark Meyer and colleagues published a study focusing on the ulnae, concluding that the morphology was more consistent with knuckle walking than with the arm function of bipedal hominins. In 2024, Marine Cazenave and colleagues published a detailed rebuttal. They systematically went through the features claimed as evidence of bipedalism and argued that none were actually diagnostic. The gluteal ridge on the femur, found in non-bipedal apes too. The femoral antitorsion, within the range of variation seen in quadrupedal primates. Cazenave's team concluded that the postcranial evidence was not sufficient to determine whether Sahelanthropus was a habitual biped. She argued that most, if not all, of the results point toward similarities with the African great apes. Then, in January 2026, a new study appeared in Science Advances that seemed to tip the scales. A team led by Scott Williams at New York University brought fresh eyes and new techniques to the analysis. Williams had not been involved in the earlier debates and approached the question with less invested in any particular outcome. His team examined high-resolution three-dimensional scans of the fossils using geometric morphometrics, comparing the Sahelanthropus bones with those of chimpanzees, gorillas, orangutans, and various fossil hominins. While analyzing the femur scan, Williams made a discovery. Rubbing his thumb along the surface of the digital bone, he felt a small bump. It was subtle, hard to see in photographs, but clearly present in the bone. This bump was located exactly where, in humans and other bipedal hominins, the iliofemoral ligament attaches to the femur. This ligament is the largest and strongest in the human body. It is loose when we are seated, but tightens around the femoral head to hold it in the hip joint when we stand and walk, preventing the torso from falling backward or sideways. The tiny bump, called the femoral tubercle, had never been reported in any non-bipedal primate. Its sole known appearance was in members of the human lineage adapted for upright walking. Jeremy De Silva, a paleoanthropologist who had previously studied the Sahelanthropus material, was startled by Williams's finding. Looking at the scans again after reading the paper, he said, "I immediately pulled this out and said, 'Wait, how did I not see this?' And sure as day, some of the key anatomies that they point out in this paper, I can see in this fossil. I'm kind of kicking myself. I wish I had seen these things." Williams's team also noted that the relative proportions of the Sahelanthropus limb bones were more human-like than ape-like. While apes have long arms and short legs, and humans have short arms and long legs, Sahelanthropus fell in between, but leaned toward the human pattern. The 2026 study did not claim that Sahelanthropus was an obligate biped, walking upright all the time like modern humans. The evidence from the ulnae still suggested significant time spent climbing. But the femoral tubercle seemed to provide the strongest evidence yet that when on the ground, Sahelanthropus walked on two legs. Even this latest study did not end all debate. Marine Kazanov responded that the femoral tubercle was in a badly preserved and damaged region of the bone. She questioned whether it was really a functionally significant structure or merely an artifact of the fossil's condition. 20 years after the discovery of Toumaï, the question of whether Sahelanthropus walked upright remained remarkably unsettled. Multiple teams of experts, examining the same fossils with increasingly sophisticated tools, continued to reach different conclusions. The controversy highlighted a fundamental challenge in paleoanthropology. When the evidence is sparse, fragmentary, and millions of years old, even the most basic questions, did this creature walk on two legs or four, can be extraordinarily difficult to answer definitively. What was clear was that the simple story told in 2002 of the oldest human ancestor definitively identified from a beautifully preserved skull had given way to something far more complex and uncertain. The femur that was supposed to have not existed turned out to exist after all. The postcranial evidence that was supposed to clarify matters had instead generated 2 decades of dispute. And the question of whether Sahelanthropus belonged on the human lineage or represented something else entirely remained very much open. Chapter 3. The Hominin or Ape Debate. Beneath all the arguments about whether Sahelanthropus walked upright lay a more fundamental question. Even if this 7-million-year-old ape did walk on 2 legs, did that make it a human ancestor? The answer was far from obvious. Classification in paleoanthropology is not simply a matter of measuring bones and checking off anatomical features. It requires making judgments about which features matter, which are primitive inheritances from ancient apes, and which are derived specializations that mark a split toward the human lineage. And for a creature as old as Sahelanthropus, living at the very edge of when humans and chimpanzees diverged, those judgments became extraordinarily difficult. Michel Brunet and his colleagues had classified Sahelanthropus tchadensis within Hominina, the group that includes humans and our direct ancestors after the split from chimpanzees. But almost immediately after the 2002 announcement in Nature, critics challenged this placement. The most pointed critique came just months later, in October 2002, also in Nature. A group of 4 researchers led by Milford Wolpoff published a comment with a provocative title, Sahelanthropus or Sahelpithecus. The question mark spoke volumes. These researchers were suggesting that instead of being an early human, Toumaï might be an early ape, deserving a name that reflected its true nature. Wolpoff and his co-authors, Brigitte Senut, Martin Pickford, and John Hawkes, laid out their objections systematically. They pointed out that the features Brunet had used to classify Sahelanthropus as a hominin were not actually unique to the human lineage. Small canine teeth, found in some fossil apes that were clearly not human ancestors. Reduced face projection, not exclusive to hominins either. The position of the foramen magnum, far from conclusive evidence of upright walking, and in any case, the opening's position overlapped with the range seen in living apes. What troubled these critics most was the mosaic nature of Sahelanthropus. The skull combined features that seemed primitive, like its small brain and massive brow ridges, with features that seemed surprisingly advanced, like the flat face and small canines. But mosaics can be deceiving. Just because a creature shares some traits with humans does not mean it is on the human lineage. Evolution is full of examples of convergence, where similar features evolve independently in unrelated lineages facing similar environmental pressures. Senut and Pickford went further, proposing an alternative identity for Toumaï. They suggested that the skull might belong to a female gorilla ancestor. The logic went like this. The skull's small size and relatively small canines could indicate a female individual. Female apes generally have much smaller canines than males, since the enlarged male canines serve primarily for display and competition with other males. If Toumaï was a female, then the reduced canines said nothing about its evolutionary position. Modern female gorillas have relatively small canines, and the face of Toumaï, with its pronounced brow ridges and overall robust construction, bore certain resemblances to gorilla anatomy. This gorilla hypothesis had interesting implications. At the time, and still today, the fossil record of gorillas was virtually non-existent. Modern gorillas live in the forests of Central Africa, environments where fossilization is extremely rare. Not a single confirmed fossil gorilla more than a few 100,000 years old had ever been found. The same was true for chimpanzees, with only a handful of teeth from about 500,000 years ago representing their entire known fossil record. If Sahelanthropus turned out to be an early gorilla relative, it would be the oldest member of that lineage ever discovered, filling a 7-million-year gap in the fossil record. The lack of fossil gorillas and chimpanzees presented a fundamental problem for interpreting Sahelanthropus. How could researchers confidently say whether a 7-million-year-old skull belonged to the human lineage, the chimpanzee lineage, or the gorilla lineage when they had almost no fossils of ancient chimps or gorillas to compare it against? The assumption underlying Brunet's classification was that Sahelanthropus looked more human-like than ape-like. But what did an early chimpanzee or gorilla ancestor actually look like? No one knew, because no one had ever found one. Bernard Wood, a prominent paleoanthropologist at George Washington University, highlighted this issue in his commentary on the discovery. Wood pointed out that the features used to identify Sahelanthropus as a hominin might not be as diagnostic as Brunet claimed. Take the small canines. Yes, reduced canines are a hallmark of later human ancestors. But several extinct apes from the Miocene, creatures clearly not on the human line, also had relatively small canines. Oreopithecus, an 8-million-year-old ape from Europe, had reduced canines and a relatively forward foramen magnum. In the 1950s, researchers had even proposed Oreopithecus as a human ancestor based on these features, along with postcranial bones that some interpreted as indicating bipedalism. But further study definitively showed it was a side branch, an extinct ape lineage that happened to converge on some human-like features. The parallel was troubling. Might Sahelanthropus be another Oreopithecus, mistaken for a human ancestor based on convergent features? The flat face presented similar problems. Brunet had made much of the fact that Sahelanthropus had a relatively flat, non-projecting face compared to living apes. This was true. But was facial flatness really a defining feature of early hominins? Some researchers argued that it might be more related to the biomechanics of chewing, to the forces generated by powerful jaw muscles acting on the skull. A robust chewing apparatus could pull the face inward, creating a flatter profile, without any connection to human evolution. This brought up a deeper issue that would haunt the interpretation of Sahelanthropus and other proposed early hominins. At the base of the human family tree, where the lineages of humans, chimpanzees, and gorillas had only recently diverged, how different would these newly separated species actually look? The last common ancestor of humans and chimpanzees was, by definition, neither human nor chimpanzee, but something ancestral to both. It probably looked more like an ape than like a human, because apes represent the ancestral condition, and humans the derived, specialized condition. So the earliest members of the human lineage after the split would still look very ape-like, because they had only just begun to evolve human characteristics. This created what some researchers called a paradox of identification. The earlier you go in time, the harder it becomes to distinguish human ancestors from their ape relatives, because the differences between them were still minimal. Yet researchers needed to make that distinction in order to interpret the fossils. The risk was circular reasoning, defining hominins as those with certain features, then using those features to identify fossils as hominins, without independent evidence that those features were actually reliable markers of the human lineage at such an early date. The age of Sahelanthropus added another layer of difficulty. At 7 million years old, the fossil stood right at the edge of when molecular biologists estimated the human-chimpanzee split occurred. Initial genetic studies had used mutation rates of about 70 mutations per generation, which placed the split between 6 and 4 million years ago. But in 2012, Adam Scally and Richard Durbin discovered that the mutation rate was actually half that, placing the last common ancestor anywhere from 14 to 7 million years ago. This revision meant that the human-chimpanzee divergence could have been significantly older than many researchers had assumed, making the timing of Sahelanthropus less problematic. But it also meant greater uncertainty about when the split actually occurred. Some researchers argued this timing actually argued against Sahelanthropus being a hominin. The species appeared too old, too close to the common ancestor, to have already developed the suite of features that later distinguished humans from apes. Critics also pointed to the geographic argument. Sahelanthropus was found in Chad, 1,500 miles west of the East African Rift Valley, where most early hominin fossils had been found, suggesting it might represent a more widespread population of apes rather than a human ancestor specifically. Yet the timing argument cut both ways. If 7 million years ago was when the lineages were splitting, then finding fossils from that time was crucial for understanding what that split looked like. Sahelanthropus might represent exactly what an early hominin should look like, mostly ape-like, but with the first tentative steps toward human characteristics. The mosaic of features was not a problem, but an expectation. The scientific literature over the following years filled with papers arguing different positions. Some researchers, analyzing various features of the Sahelanthropus teeth and jaw, concluded it was indeed a hominin. Others, looking at the same features, concluded it was an ape. A particularly detailed analysis published in 2006 by Wolpoff, Hawkes, Sanute, Pickford, and a fifth colleague, James Ahern, laid out their full case under the title, An Ape or the Ape? Is the Toumaï cranium TM266 a hominid? This paper systematically went through the anatomy. The authors measured the Toumaï skull and compared it with chimpanzees, gorillas, and various fossil hominins. They concluded that in most respects, Toumaï looked like a female gorilla. The overall shape of the cranium, the structure of the brow ridges, the way the back of the skull was constructed, all of this fit better with gorillas than with known hominins. They even claimed to see evidence that the canine teeth had wear patterns consistent with the blade-like sharpening that occurs in apes when the upper canine rubs against the lower premolar. Brunet had said this wear pattern was absent in Sahelanthropus, marking it as human-like. Wolpoff's team said it was present, marking it as ape-like. Brunet fired back, defending his classification vigorously. In a reply published in Nature, he and his team argued that the critics had misidentified features and misunderstood the anatomy. The supposed ape-like wear patterns did not exist when you examined the actual teeth carefully. The features that Wolpoff claimed were gorilla-like were actually within the range of variation seen in early hominins. Brunet noted that the critics' alternative interpretation was not supported by published or unpublished data and suggested their bias was showing. The implication was clear. Sanute and Pickford were the discoverers of Orrorin tchadensis, which they had claimed was the oldest hominin before Sahelanthropus was found. Professional rivalry, Brunet implied, was coloring their scientific judgment. In 2011, Bernard Wood returned to the question with a major review article in Nature co-authored with Terry Harrison. They argued that three of the main features used to identify early hominins, small canines, forward-positioned foramen magnum, and various aspects of postcranial anatomy, were actually not reliable diagnostic characters for the earliest members of the human lineage. All three features could be found in fossil apes that were definitely not hominins. The Oreopithecus example stood as a stark warning against overinterpreting such features. Wood and Harrison suggested that Sahelanthropus, Orrorin, and Ardipithecus, the three oldest proposed hominins, were all questionable. None of them had been definitively proven to be on the human lineage rather than on the chimpanzee lineage, or representing extinct ape lineages that split off before the human-chimp divergence. The evidence was simply too ambiguous, and the features used to classify them were not specific enough. This critique struck at the heart of how paleoanthropology worked. If you could not trust small canines, foramen magnum position, or even postcranial adaptations as reliable markers of hominin status in the earliest species, then what could you trust? How could you ever identify the oldest human ancestors? One possible answer was that you could not with certainty, not until you had a much richer fossil record that showed clear transitional sequences and unambiguous specializations unique to the human lineage. The evidence was simply too ambiguous, and the features used to classify them were not specific enough. This critique struck at the heart of how paleoanthropology worked. If you could not trust small canines, foramen magnum position, or even postcranial adaptations as reliable markers of hominin status in the earliest species, then what could you trust? How could you ever identify the oldest human ancestors? One possible answer was that you could not with certainty, not until you had a much richer fossil record that showed clear transitional sequences and unambiguous specializations unique to the human lineage. Some researchers pushed back against this pessimism. They argued that while individual features might not be diagnostic in isolation, the combination of features in Sahelanthropus was. No fossil ape had the exact same mosaic as Toumaï. The particular way that small canines, facial structure, and cranial base configuration came together pointed toward hominin status, even if none of those features alone was conclusive. The question of whether Sahelanthropus was a hominin also connected to broader debates about what it even means to be a hominin. The traditional definition focused on bipedalism. Hominins were the bipedal apes. But if bipedalism evolved multiple times in different lineages, or if some early hominins were not yet fully bipedal, then that definition became problematic. An alternative definition focused on the evolutionary relationship. Hominins were all species more closely related to humans than to chimpanzees. But this was a phylogenetic definition that required knowing the evolutionary tree, which was precisely what was in dispute. The classification debate took on new dimensions when postcranial material finally entered the discussion in the 2020s. As detailed in the previous chapter, the existence of a femur and other limb bones found with the Toumaï skull became one of the most contentious issues in modern paleoanthropology. Different teams of researchers examining the same fossils with increasingly sophisticated techniques continued to reach opposite conclusions about whether the postcranial evidence supported habitual bipedalism or suggested a more ape-like mode of locomotion. By the 2020s, the classification of Sahelanthropus remained officially uncertain. Different researchers and different textbooks placed it differently. Some phylogenetic analyses positioned it as the earliest hominin on the human lineage. Others positioned it as a stem hominin, closer to both humans and chimps than to gorillas, but before the human-chimp split was complete. Still others placed it as an early gorilla relative, or simply as an extinct ape lineage of unknown affinity. What everyone agreed on was that Sahelanthropus was important. Whether it turned out to be a human ancestor, a gorilla ancestor, or neither, it was a 7-million-year-old African ape from a crucial time period. It provided a rare glimpse into what apes looked like right around the time when the lineages leading to modern humans, chimps, and gorillas were diverging. The fundamental classification debate over Sahelanthropus highlighted the limits of science when dealing with fragmentary evidence from the deep past. A single distorted skull, a disputed femur, 2 partial ulnae, a handful of teeth, and jaw fragments. This was the entire evidentiary basis for deciding whether a species belonged to our lineage or to another. Researchers could argue endlessly about the interpretation of these bones because the bones themselves did not carry clear answers. They required interpretation, and interpretation required assumptions, and different assumptions led to different conclusions. More than 2 decades after its discovery, Sahelanthropus tchadensis remained at the center of one of paleoanthropology's most enduring controversies. The question was not simply academic. If Sahelanthropus was a hominin, it pushed the human lineage back to at least 7 million years and suggested our origins were geographically broader than once thought. If it was not a hominin, then the search for our oldest ancestor continued, and the nature of that ancestor remained uncertain. The classification of this one skull had implications for the entire story of where we came from and how we became human. And as the ongoing scientific debates demonstrated, that story was far from finished being written. Chapter 4. The Silverback's Domain. Roberto Macchiarelli, who had been recruited from Rome to join Michel Brunet's research group at the University of Poitiers, would later describe his colleague with a vivid metaphor. Michel is a dominant male. He's a silverback gorilla. The comparison was apt in more ways than one. In the social structure of gorilla groups, the silverback controls access to everything: resources, mates, territory. Challenge the silverback and face the consequences. Brunet ran his research group at Poitiers with a similar intensity, and the discovery of Sahelanthropus only amplified dynamics that had been in place for years. The laboratory was organized around Brunet's projects, his priorities, his vision. Other scientists in the group were expected to provide what one researcher called service to Brunet's work rather than pursue independent lines of inquiry. Graduate students and junior researchers could benefit from association with his high-profile discoveries and the resources he commanded: prestigious publications, access to grant funding, the chance to work on fossils that made headlines around the world. But there was a price: deference. Brunet had the authority to approve or deny projects undertaken by members of his group. If a proposed study did not advance his own research agenda, he would refuse to sign off on it. This tight control meant that alternative perspectives, questioning voices, were not always welcome. For some researchers, this arrangement was tolerable. The prestige of working on Toumaï outweighed the constraints. But for others, particularly those with established reputations of their own, Brunet's expectation of loyalty grated. Olga Otero, who worked with Brunet for years, observed that he seemed to treat the Sahelanthropus fossils as if they were the most precious thing on Earth, assuming that everyone, everywhere, must be consumed with the desire to study them. This possessiveness extended beyond simple scientific caution into something more personal, more territorial. The fossils were not just specimens to be studied, but symbols of achievement, markers of status, property to be controlled. For Macchiarelli, who was 15 years Brunet's junior but had no interest in playing the role of subordinate, the tensions became untenable. When Macchiarelli publicly stated that doubts, additional questions, argument, and criticism contribute to the strength, not the weakness of scientific thought, it was both a general principle and a pointed rebuke. Brunet responded by referring to him dismissively as the Italian, a label that managed to be both diminishing and xenophobic. The professional relationship deteriorated into a personal animosity that would poison scientific discourse around Sahelanthropus for years to come. The conflicts within Poitiers were mirrored by disputes that extended far beyond the university walls. The discovery of Sahelanthropus had not unfolded quite as Brunet's carefully crafted public narrative suggested. The version presented to the world in press conferences and scientific papers was that Brunet's expedition team had discovered the fossils during fieldwork he directed in Chad. Brunet was the leader, the director, the discoverer. But the reality was more complicated. Alain Beauvillain, a French geographer who handled logistics for the Chad expeditions, had a different version of events. According to Beauvillain, the July 2001 trip that yielded Toumaï was not actually a Brunet-led expedition at all. It was an impromptu prospecting mission that Beauvillain organized with 3 Chadian colleagues while Brunet was back in France. Beauvillain and his team, Ahaunta Jimdumalbai, Fanone Gongdibe, and Mahamat Adoum had set out from Jamena without Brunet's explicit authorization. When Jimdumalbai spotted the skull on July 19th, Brunet was not present. He was not even in Chad. In the conventions of paleoanthropology, the director of a research program is credited as the discoverer, even if not physically present when a fossil is found. The logic is that the director organized the expedition, obtained permits, secured funding, and provided the framework that made the discovery possible. By this standard, Brunet's claim to have discovered Sahelanthropus was legitimate. But Beauvillain chafed at how thoroughly he and the Chadian team members were written out of the story. In interviews and articles, Beauvillain made sure to set the record straight. He had organized this particular trip, and Jimdumalbai had actually spotted the skull.
Brunet did not take kindly to this alternative narrative. He and Beauvillain had already been feuding before the Toumaï discovery, and this public airing of who really found the skull intensified the conflict. As soon as the description of Sahelanthropus was published in Nature in 2002, Brunet had Beauvillain recalled from Chad back to France. The message was clear: challenge the official story and pay the price. In subsequent accounts, Beauvillain's role was minimized or erased. The Franco-Chadian paleoanthropological mission's list of team members often omitted his name. The narrative belonged to Brunet, and Brunet would control how it was told. This pattern of controlling the narrative extended to how the fossils themselves were managed. After the initial study period in France, the Sahelanthropus remains were returned to Chad, where they were housed at the National Museum in Jamena. But the original fossils were kept under tight security, not available for general examination. Instead, plaster casts were put on public display. When other scientists wanted to study the material, they had to seek permission, and such permission was not automatically granted.
The relationship between Brunet and the Chadian government became a crucial element in maintaining this control. Brunet had cultivated a close friendship with President Idriss Déby, who had personally chosen the name Toumaï for the skull. The Chadian government had embraced Sahelanthropus as a source of national pride. The national airline had been renamed. The fossil appeared on stamps and currency. Brunet had become a celebrity in both France and Chad, receiving major awards and recognition. This political capital gave him influence over who could access the fossils and under what conditions. The arrangement served both parties. Chad gained international prestige, and Brunet gained control over a discovery that could make or break scientific reputations. The issue of access became particularly contentious when plans were made to tour the Sahelanthropus fossils in the United States. In 2007, a traveling exhibition called Lucy's Legacy, The Hidden Treasures of Ethiopia, was being organized by the Houston Museum of Natural Science, featuring not just the famous Lucy skeleton of Australopithecus afarensis, but also other significant fossil hominins, including Sahelanthropus. The exhibition would tour 10 American museums over 6 years, and a portion of proceeds would go to modernizing Ethiopian and Chadian museums. But the decision to allow the fragile, irreplaceable original fossils to travel raised immediate concerns in the scientific community.
Several prominent researchers publicly opposed the tour. Owen Lovejoy, a paleoanthropologist who had worked on reconstructing the Lucy skeleton, stated his opposition based on the risk of damage. Richard Leakey, the legendary fossil hunter and anthropologist, also spoke against it. These fossils were unique. If something happened to them during transport or while on display, the loss to science would be catastrophic. Several major museums declined to host the exhibition at all. The Smithsonian Institution passed. The Cleveland Museum of Natural History, which had previously housed and studied the Lucy fossils, also declined. Their reasoning centered on the risk to the specimens, but there was also discomfort with what some saw as treating priceless scientific material as a traveling show. Most museums that display hominid fossils use casts, not originals, for exactly this reason. The originals are too valuable to risk. Even Donald Johanson, who discovered Lucy and might have been expected to support such exhibitions as a way to educate the public about human origins, expressed concerns about the fragility of the specimens, though he ultimately felt the educational benefits might outweigh the risks. But the message from much of the scientific community was clear. This was a bad idea. Yet the exhibition went forward anyway, approved by the Ethiopian and Chadian governments and the U.S. State Department. The fossils went on tour from 2007 to 2013, traveling to Houston, Seattle, New York, and other cities before finally being returned. This willingness to transport the originals for a public exhibition stood in stark contrast to how access was controlled for scientific research. While thousands of tourists could file past the fossils in glass cases, paleoanthropologists who wanted to examine them closely for research purposes faced more restrictions. The fossils were Brunet's to manage, in collaboration with the Chadian government, and that management reflected his priorities. The most explosive battle, involving a femur found with the skull and kept unpublished for nearly 2 decades, became a case study in how control over fossils could shape scientific conclusions. The rivalry also intensified because critics like Brigitte Senoot and Martin Pickford had their own competing early hominin, Orrorin tchadensis, giving professional motivations to their challenges. Even the 2026 discovery by Scott Williams of a femoral tubercle suggesting bipedalism could not end the debate, as questions about the fossil's preservation immediately arose. These battles revealed deeper problems about how spectacular fossil discoveries were managed in paleoanthropology. The incentive structures of the field, the fame and funding that come with discovering a major new hominin, the competition between research teams and nations, these forces shaped behavior regardless of individual personality. When a single skull could make or break careers, when national pride got wrapped up in fossils, when institutions competed for the prestige of hosting exhibitions, the science risked being overwhelmed by everything around it.
The questions that should have mattered, what was this creature, how did it move, where did it fit in the story of evolution, got tangled up with questions of credit, ownership, reputation, and power. Brunet was perhaps an extreme case of territorial control, but he was not unique. The field had seen similar battles over other major discoveries. Disputes over who deserved credit, accusations of stolen data, restricted access to crucial specimens, personal animosities that lasted decades. What the Sahelanthropus controversy highlighted was how much the scientific process depended on norms of transparency and evidence sharing that were enforced only by social pressure and professional reputation. There were no formal mechanisms to compel researchers to publish findings or share access to fossils in their control. The specimens technically belonged to the countries where they were found, but in practice, the scientists who excavated them controlled them. If a researcher chose to withhold evidence, delay publication, or restrict access, there were limited remedies available to the scientific community. The toll this took on scientific progress was significant. 2 decades after the discovery, researchers were still arguing about basic facts, unable to reach consensus on whether Sahelanthropus was bipedal, whether it was a hominin, or what it meant for understanding human origins. The tragedy was that this was not how science had to work. In other parts of paleoanthropology, researchers with competing interpretations managed to engage in civil debate, sharing data and working toward resolution even when they disagreed. With Sahelanthropus, that commitment seemed compromised.
The academic rivalries and battles over access had created an environment where trust broke down, where every claim was viewed with suspicion, where the sharing of information became a strategic calculation rather than a scientific obligation. Brunet's dominance, his silverback control over the fossils and the narrative, meant that alternative voices struggled to be heard. The result was a controversy that might have been resolved years earlier with more open access and collaborative spirit. By the 2020s, as new analyses continued to emerge and debates continued to rage, the scars from these battles remained deep. The scientific community had learned, or should have learned, that the way fossils are controlled matters as much as the fossils themselves. Great discoveries require great scientists, but they also require institutions and norms that prevent any single individual from becoming the sole gatekeeper to evidence that belongs to all of humanity. Whether Sahelanthropus ultimately proved to be a human ancestor or not, the story of how it was fought over had become inseparable from the science itself, a cautionary tale about power, personality, and the fragile social foundations on which the search for our origins rests.
Chapter 5. Beyond the Rift. For most of the 20th century, paleoanthropologists could draw a map of human origins with a relatively simple line. The map would show Eastern Africa, specifically the Great Rift Valley, that massive geological scar running from Ethiopia south through Kenya and Tanzania. Along this valley, researchers had found fossil after fossil, building up a picture of human evolution that seemed to tell a coherent geographic story. The earliest members of the genus Australopithecus came from sites like Laetoli in Tanzania and Hadar in Ethiopia. The earliest members of the genus Homo came from Olduvai Gorge in Tanzania and Kubifora in Kenya. The progression seemed clear. Humans evolved in the rift, and for millions of years, that narrow strip of eastern Africa was the stage on which our species' drama unfolded. This geographic concentration of fossils led to a theoretical framework that became known as the East Side Story. The name was a playful reference to the musical West Side Story, but the hypothesis behind it was serious scientific work, particularly associated with the French paleontologist Yves Coppens. Coppens proposed that the formation of the Great Rift Valley itself had been the driving force behind human evolution. According to this model, the geological processes that created the rift, beginning around 8 million years ago, did more than just crack open the earth. They created a barrier that split populations of ancient apes and fundamentally altered the climate on each side. To the west of the rift, the reasoning went, conditions remained relatively unchanged. Moist air from the Atlantic Ocean continued to bring rain. Forests persisted. The apes living in these western forests could continue their arboreal lifestyle, swinging through the trees, feeding on fruit, moving on all fours when they descended to the ground. This western population, the theory suggested, gave rise to modern chimpanzees and gorillas, creatures that retained the ancestral ape body plan and way of life. But to the east of the rift, everything changed. The uplifted highlands of the Rift Valley created a rain shadow. Moisture-laden winds from the west dropped their rain on the western slopes, leaving the eastern side progressively drier. Over millions of years, the lush forests thinned out. Grasslands expanded. Woodlands became more open. The apes living on this eastern side faced a new challenge: how to survive in an increasingly open landscape where food was more scattered, and the safety of continuous tree cover was lost. The East Side Story proposed that this environmental pressure drove the evolution of bipedalism. Walking upright on two legs is energetically efficient for covering long distances across open ground. It frees the hands to carry food from one scattered resource patch to another. It raises the eyes higher, allowing better surveillance for predators in grassland environments where visibility matters. The apes that adapted to this new lifestyle by becoming bipedal were the ones that survived and flourished on the eastern side of the rift. They were our ancestors. This narrative was elegant, geographically specific, and supported by an impressive array of fossil evidence. Every confirmed hominin fossil older than 3 million years had come from eastern or southern Africa, and the vast majority from the Rift Valley itself. When researchers mapped the distribution of fossil sites, they created a clear pattern. The Rift was special. It was not just where humans evolved, but the reason humans evolved, the geographic and climatic crucible that forged our lineage. The discovery of Sahelanthropus in Chad, 1,500 miles west of the Great Rift Valley, immediately challenged this framework. If a 7-million-year-old potential hominin lived in Chad, far from the supposed evolutionary crucible, then the Rift Valley Formation could not have been the trigger for human evolution. The geographic implications were profound and unavoidable. The distance involved was enormous. Chad is separated from the Rift Valley by more than 1,000 miles of what is now some of the most inhospitable terrain on Earth, including much of the Sahara Desert. 7 million years ago, the Sahara did not exist in its current form. The region was wetter, more habitable. But still, the expanse was vast. Was Sahelanthropus an isolated Western population cut off from relatives in the East? Or were early hominins actually distributed across a much broader swath of Africa than anyone had imagined? Interestingly, this possibility had been anticipated even before the Toumaï discovery. In 1998, an anthropologist named Noel Boaz had proposed, contrary to the prevailing rift theory, that the ancestral population giving rise to both chimpanzees and humans might have become isolated in a riverine forest zone in Chad, surrounded by more arid open lands. Later, according to Boaz's speculation, a forest corridor could have allowed movement into East Africa. At the time, this was just a hypothesis with no direct fossil evidence. The discovery of Sahelanthropus 3 years later suddenly made Boaz's speculation look prescient. Some researchers, looking for ways to reconcile the Chad discovery with the East Side story, suggested that perhaps the human lineage did evolve in the East but then spread west, and Sahelanthropus represented that westward expansion. But this explanation had problems. If Sahelanthropus was really the oldest hominin, older than anything found in the Rift Valley, then the expansion would have had to happen before there were any clear hominins in the East. That made no sense. The simpler explanation was that human evolution was not an East Side story at all. It was a Pan-African story, or at least a story that played out across a much larger stage than the narrow Rift Valley. Understanding where Sahelanthropus lived required reconstructing an ancient environment from fragmentary clues. The Jurab Desert today is one of the most desolate places on Earth. But 7 million years ago, it was utterly different. Analysis of thousands of fossils from the site, including fish, crocodiles, mammals, and primates, revealed a mosaic environment with lakes, gallery forests, and grasslands, very different from the open savanna predicted by East Side story. Analysis of the plant-eating mammals, particularly the bovids, provided crucial insight. Researchers examined microscopic wear patterns on the teeth of fossil antelopes and related species from the site. Different types of vegetation create different wear patterns. Grass, with its high silica content, creates distinctive scratches. Leaves from trees and shrubs create different patterns. By analyzing these patterns, scientists could infer what the animals were eating, and by extension, what vegetation was available in the environment. The bovid teeth from Toros Manala showed a mixed signal. Some species seemed to be eating mostly grass, others mostly browse from trees and shrubs, and many showed evidence of a mixed diet. This pointed to what ecologists call a mosaic environment, a patchwork of different habitat types. There were open grasslands, yes, but also woodlands, gallery forests along rivers, and probably thickets and scrub. This was not the open savanna that the East Side story predicted should drive human evolution. It was not the continuous forest where apes supposedly remained unchanged. It was something in between, a varied landscape offering diverse food sources and diverse challenges.
The picture that emerged was of a vast lake, probably an ancient precursor to Lake Chad, surrounded by this mosaic of habitats. Modern Lake Chad, even during wetter periods in recent history, has been enormous. During the Holocene humid period around 9,000 years ago, a megafauna lake covered more than 350,000 square kilometers, an area larger than the British Isles. The ancient lake that Sahelanthropus knew might have been similarly vast, with shifting shorelines, seasonal floods, permanent marshes, and gallery forests reaching inland along rivers that fed the lake. This environment raised important questions about the role of habitat in driving human evolution. The old savanna hypothesis, closely related to the East Side story, held that the transition from forest to open grassland was the key selective pressure. Our ancestors had to adapt to living on the ground in open country, and this drove the evolution of bipedalism, larger brains, tool use, and all the other hallmarks of humanity. But Sahelanthropus lived in a mosaic environment, not pure savanna. This suggested a different model for the origins of upright walking. Perhaps bipedalism was not an adaptation to treeless grasslands, but an adaptation to moving between scattered patches of forest and woodland. In a mosaic environment, the ability to walk efficiently on the ground between resource patches while retaining the ability to climb into trees for food and safety would be advantageous. The environmental reconstruction of the Toros Manala site also had implications for understanding early hominin diets. The old model assumed early human ancestors shifted from the fruit-heavy diet of forest apes to a more varied diet, including tougher foods like roots, tubers, and seeds from grassland environments. But in mosaic environments, plenty of tough plant foods are available: hard nuts, fibrous roots from wetland plants, seeds from woodland trees. What the Chad discovery ultimately challenged was not just the geographic specificity of the East Side story, but the entire linear, deterministic narrative it represented. According to that narrative, there was a clear sequence. Rift formation led to climate change. Climate change led to habitat transformation. Habitat transformation led to bipedalism. And bipedalism led inexorably down the path to humanity. It was a story with a clear beginning, middle, and end, with each stage causing the next in predictable fashion. The reality suggested by Sahelanthropus and subsequent discoveries was far messier. Human evolution did not unfold in one place, along one pathway, driven by one environmental change. It happened across a broad region of Africa, in diverse environments, with multiple hominin species experimenting with different adaptations at different times. Some of these experiments succeeded, others failed. Some led toward later humans, others to extinction. The story was not a straight line, but a bush, branching and rebranching. By 2024, 50 years after the methods of molecular biology first allowed scientists to estimate when humans and chimpanzees diverged, and 23 years after the discovery of Toumaï, the landscape of human origins looked very different from how it had appeared in the 20th century. Johannes Haile Selassie, director of the Institute of Human Origins at Arizona State University, captured the new understanding. We have multiple hominin species in the same time period. The age of Sahelanthropus was no longer a time of lonely pioneers, a single lineage groping its way toward humanity in the East African Rift. It was a time of diversity, experimentation, and geographic spread. Researchers now understood that the environment Sahelanthropus inhabited was not an anomaly, but probably typical of what early hominins experienced. Subsequent studies of other early hominin sites, including those of Orrorin tchadensis in Kenya and Ardipithecus ramidus in Ethiopia, revealed similar mosaic environments. These creatures lived in settings with woodland, bushland, and grassland mixed together. They were not pure savanna dwellers, and their adaptations reflected the varied demands of such habitats.
The collapse of the simple savanna hypothesis for human origins raised new questions about why bipedalism evolved in the first place. If not as an adaptation to open grasslands, then what? Various alternatives had been proposed over the years. Perhaps upright walking was an adaptation for wading in shallow water at lake margins, freeing the hands to gather aquatic plants or catch fish. Perhaps it was a feeding strategy for reaching fruits in low trees and shrubs. Perhaps it was about efficient long-distance walking to visit scattered resources. Perhaps it was about keeping the body cooler under the tropical sun by minimizing the surface area exposed to direct overhead sunlight. Or, perhaps, as some researchers suggested, it was about freeing the hands to carry food back to mates and offspring, facilitating a shift toward food sharing and pair bonding. Each of these hypotheses had evidence for and against it, and the truth might involve elements of several. What Sahelanthropus contributed to this debate was evidence that whatever drove bipedalism, it was not the simple transition from forest to savanna. The mosaic environments of the late Miocene were complex settings that could have favored bipedalism for multiple reasons, and different populations in different regions might have been responding to somewhat different pressures.
The geographic challenge posed by Sahelanthropus also forced researchers to think differently about how human evolution fit into the broader context of African ape evolution. If early hominins ranged from Chad to Kenya, spanning nearly 2,000 miles, what about early gorillas and chimpanzees? Where did they live during this same period? The fossil record remained frustratingly silent. Not a single confirmed fossil gorilla or chimpanzee older than a few 100,000 years had ever been found. This meant that all discussions of what was hominin and what was ape, what was on our lineage and what was on the chimpanzee or gorilla lineage, were taking place in a profound information vacuum about what the other lineages actually looked like. The discovery in Chad demonstrated that the fossil record of human origins remained desperately incomplete. Vast regions of Africa had barely been explored for ancient fossils. What might lie buried in other unexpected places? The Sahara itself, now mostly inaccessible to paleontological work, was once a land of lakes and rivers. The forests of Central Africa, where gorillas and chimpanzees now live, had probably been occupied by apes for millions of years. But forest environments rarely preserve fossils. The geological processes that make the East African Rift so rich in fossils, volcanic activity creating datable ash layers and rapid sedimentation preserving bones, do not operate everywhere.
The East Side story had been appealing partly because it made sense of why so many fossils came from one region. But that concentration of fossils might say more about where fossils are likely to be preserved and discovered than about where human ancestors actually lived. The lesson of Sahelanthropus was that absence of evidence was not evidence of absence. Just because hominins had not been found across a broader region did not mean they had not lived there. The cradle of humanity was bigger than researchers had thought, more diverse in its environments, and more complex in its evolutionary dynamics. Multiple lineages of apes were experimenting with different adaptations across a broad region of Africa during the late Miocene. Some of those experiments led toward modern apes, others toward humans, and many led nowhere at all. The story was not one of linear progress driven by a single environmental shift in a single location. It was a story of diversity, contingency, and the unpredictable branching of evolutionary possibility across a vast and varied continent.
The story of Sahelanthropus tchadensis reveals that the search for human origins is as much about the scientists as the science. 25 years after Toumaï's discovery, fundamental questions remain unresolved. Was this 7-million-year-old ape our ancestor, or something else entirely? Did it walk upright or move like modern apes? These debates persist not just because the evidence is fragmentary, but because of how fossils are controlled, how rivalries shape interpretation, and how the incentives of the field sometimes work against open collaboration. What's clear is that human evolution was not a simple story unfolding in one place.
The geographic breadth, environmental diversity, and species plurality of our origins demand humility about what we think we know. Each new discovery doesn't just fill gaps in our knowledge, it reveals how much territory remains unmapped. Perhaps the deepest lesson of Sahelanthropus is this: the bones themselves hold secrets, but understanding them requires not just sophisticated technology and careful analysis, but also the willingness to share evidence, embrace uncertainty, and recognize that the story of where we came from belongs to everyone. Thank you for listening.